Okay, so we have transcription, translation in prokaryotic cells, and then we have transcription and RNA processing and translation. So yesterday in the video I talked about these two steps here with transcription, how exactly we make an mRNA molecule. So remember that you have the enzyme called, it's involved with the enzyme. It's RNA polymerase, sorry. So function's very similar to DNA polymerase. DNA polymerase is used in DNA replication to make new DNA strands. So RNA polymerase is used to make RNA from the DNA. So what happens, remember, is that RNA polymerase separates the two DNA strands, and we only use one side of the DNA because RNA is single-stranded, and RNA is built in the 5' to 3' direction, all right, with complementary base pairs of the DNA, and that's called your pre-mRNA. And then we go through and we process it, which takes two steps, which is cutting out. What do we cut out? What do we call the parts that we cut out? Introns, and the exons are what we keep. So, so this is this. So here we have. That's where you have, here's your gene, so this is your DNA molecule, gene, beginning and end. We have exons and introns. So our processing, this is the DNA, so transcription is making an mRNA strand from that. So the mRNA starts at the beginning of the gene and goes to the end of the gene. But that mRNA still has exons and introns here, the same exons and introns that the DNA has. So then RNA processing, so this is the pre-mRNA, I should say, pre-mRNA. And then RNA processing is cutting out those introns and putting together the exons. So you have the appropriate amino acids, so they can be read in codons in groups of 3, and it brings one amino acid hooked to another amino acid, and so on and so forth. And so we have to do that in eukaryotic cells in order to make the right protein. If we left the introns in, we'd have additional codons, and therefore we have additional amino acids, and you get a different protein, all right? And so making the right protein it really involves cutting out the right introns. And so what I left off with in the video is that different organisms can have the same gene but make different proteins based upon what they treat as introns and exons. So can you see that if we did not treat this as an intron, if we left this and treated it as an exon, so instead of this being an intron, this was the exon, that means that when we cut and do the RNA processing, we wouldn't splice this out, we wouldn't cut this out, we just cut this one out. So now we have additional bases, and we have additional then amino acids, and then you can make more proteins, and so, or different proteins as a result of that. Let me drink this. So not only with different organisms that can do that, but also in us. And I used the example that we actually make way more proteins than we have genes. And if one gene made one protein, you have equal numbers for every gene, one protein. So they've done a lot of studying to figure out how in the world with only X amount of DNA and X amount of genes, how do we make so much protein? And they found out that sometimes we treat what's an exon and what's an intron differently. So if we treat this as an intron and cut it out and put these exons together, we get one protein. But then if we take that same piece of DNA and then treat this as an exon, then we get a different protein, and so on and so forth. So from one stretch of DNA from one gene, even though it's the same piece of DNA, we can make multiple proteins from that same gene, depending upon what we treat as an intron and what we treat as an exon. Does that make sense? Okay. All right, so that's where we're at. So the last thing we need to look at is translation, is actually how we build the polypeptide chain. Translation is when the cell interprets the genetic message and builds the polypeptide. tRNA acts as the interpreter. It transfers amino acids from the cytoplasm to the ribosome, where they are added to the growing polypeptide. And all tRNA molecules are different. So, we've talked about, this is your cell, here's your nucleus, so here's your DNA, and there's a gene as part of the DNA, and then we go through transcription where we make our pre-mRNA strand, and then we go through RNA processing, splicing out the the introns, and so we have a smaller mRNA strand. And so that's what we've talked about. Now that mRNA leaves and is going to go to the ribosome, and that's where translation is going to occur. So we're gonna read the mRNA in groups of 3 called codons at the ribosome and bring amino acids and hook them together to make the appropriate protein. And so this tRNA molecule is instrumental in that. So, so tRNA structure and function. So tRNA, like mRNA, is made in the nucleus and is used over and over again. tRNA has an amino acid. AA is amino acid, so you might want to write that out if you don't. I think you're going to remember at one end, and an anticodon at the other. The anticodon acts to base pair with the complementary code on the mRNA molecule. Let me talk about this picture here and what this means. So tRNA, like mRNA, is made in the nucleus and is used over and over again. So my board here is the nucleus. So here's our DNA. And we've talked that you have a gene is a section of that DNA. And so when we go through transcription from that gene, we make an mRNA strand. And go through processing and so on. And then use eventually that mRNA to hook together amino acids. So this is your polypeptide chain. So this is the whole thing. So some genes code for making a protein by first coding for an mRNA strand by doing the complementary bases and then bringing with it the appropriate polypeptide. And so, so if your DNA, let's say, let's say the first 3 bases is UAC on one side of the DNA, the side of the DNA that's going to be used to make the mRNA. What would be the first 3 bases then of the mRNA? When this is so DNA is double-stranded, we're going to use one side. Remember, it's built in the 3. The DNA, we read it in the 3 to the 5' end, so the mRNA is built in the 5 to 3' end. So this, when we build this, we're going to use the complementary bases. So you should have A, what goes with the U, A, because what goes with the A, it's U, because this is RNA, so it's not DNA. It would be T if it was DNA, but it's not. All right, and so we have AUG. And then, yes?
That DNA is.
Oh.
Yeah.
All right, sorry. T. Thank you for catching me. All right, it's supposed to be TAC, so then we have AUG. And then the first amino acid then would be, what do we use in we use our chart, right? The chart that we learned about yesterday. So when I use the chart, what am I using? Am I going to use TAC or AUG? You use AUG. So you always use what's on the mRNA codon on the chart. So if you flip to your chart and you look at AUG, there's a few pages back there. AUG, you should see that it says start, or it says, actually brings, stands for an amino acid, and it's MET, right? M-E-T. So what you got?
Yep.
Now on this chart, you may want to write in big letters above it, use mRNA codon. Just to remind you to use the mRNA in that chart and not try to use something else, okay? Use the mRNA. So now, how does tRNA, what is this tRNA thing going on? So tRNA is another type of RNA molecule. Different than mRNA, it has a different job. So what I want to point out here is that some genes code for making mRNA, which directly is involved in protein synthesis, but some genes don't actually code for making a polypeptide. So let's say we have a gene here. This gene is used to make tRNA. And that's it. The tRNA doesn't code for making a polypeptide. So this gene, the sole purpose is to be used to make tRNA. Now what tRNA is a chain of RNA nucleotides like mRNA. And what they do is they fold on themselves and get a three-dimensional shape. So when I say that this line here is going to be this tRNA molecule here, so it folds on itself. Two-dimensionally, it kind of looks like a clover shape. RNA is single-stranded, so it has A's, U's, C's, and G's that aren't bonded to anything. So when they fold on themselves, A's can bond with U's and C's bond with G's. And so you have this three-dimensional shape. So what we're looking at now is, so it's not a straight line. It has two parts to it. It has an amino acid attachment site, and it has an anticodon. Anticodon means it's opposite of the mRNA's codon, or has the complementary bases, to mRNA, the mRNA codon. Alright, so that's why they call it an anticodon. So we're talking about the codon on mRNA. So then three-dimensionally, what this looks like is this. So sometimes they say that this looks like the kind of an upside down, backwards letter, capital letter L, has this particular shape here. So this is the three-dimensional shape, but here's your anticodon region and your amino acid binding site. So what happens is that there are lots of different tRNA molecules that base pair with the mRNA's codon. So for instance, when we say that your DNA is TAC, we make an mRNA, the first codon is AUG, and when we use our chart, we say, okay, the first amino acid is going to be methionine. So how does the first amino acid get there to be methionine? And this is because, so we want, if we look at the big picture here, we have our first codon here, AUG, and we want the amino acid methionine to come over here and be that first amino acid. So what gets it there? And that's what these tRNA molecules are. These tRNA molecules have these anticodons, and this is what it looks three-dimensionally, but our textbook, makes it look like this computer-generated image here. So this is the tRNA molecule with the amino acid site and the anticodon. So what happens is that we have tRNA molecules floating around in our cytoplasm, all right, and not attached to any amino acids. So what happens is that we have also amino acids floating around in our cytoplasm. Why do we have amino acids in our cytoplasm? Where would those come from? How would we get amino acids to build proteins in our cells? Our diet. All right? Our diet. We eat protein. So you eat a hamburger. I've talked about this when we talked about the macromolecules. You eat a hamburger, That's cow protein, is a bunch of amino acids hooked together. When that hamburger goes through your digestive system, you digest that protein into individual amino acids. Those amino acids that were once all hooked together to make the cow protein now go into your bloodstream and get carried to all the cells of your body. And now those amino acids that used to be all put together to make the protein of a cow, now we can use to make human proteins and be used to build our own proteins. And so how did the right amino acid get to be the first one? So how does methionine get to be the first amino acid? And it's the help of these tRNA molecules. So therefore, why AUG stands for methionine in our chart, why that is because we have floating around in our cytoplasm, we have tRNA molecules that have an anticodon with the opposite bases of the mRNA. So therefore, if the first codon is AUG, what would be the anticodon that would bind to that? It's going to be T?
No.
Oh, one of my mistakes there. The T. It's RNA. This is tRNA. So it's going to be, so what goes with an A? A U. What goes with a U? An A. And what goes here? A C. And so therefore, we have here, I'm going to put here the anticodon UAC right here. If the anticodon is UAC, notice that then, so here's my my mRNA, AUG. So now I have a tRNA molecule floating around in my cytoplasm with the anticodon UAC. So what happens is this particular tRNA molecule picks up and binds to a specific amino acid. So this is the amino acid attachment site. And so, so what amino acid this tRNA picks up is methionine. So this tRNA molecule with the anticodon UAC always will pick up methionine. So here we have, it'll pick up that particular amino acid. And remember, there are 20 different amino acids. So there are each tRNA molecule only picks up one kind. So that tRNA molecule with UAC can't pick up any of the other 19 amino acids. It only can bind with methionine. So therefore, this tRNA molecule with this anticodon always brings that amino acid. But notice here that this anticodon is the only one that will bind to the mRNA with the AUG. So if you had a tRNA with CCA, it won't bind there, and therefore that amino acid that tRNA is carrying won't be the first amino acid. So therefore, with the mRNA codon, only one tRNA can bind to it, the tRNA with UAC, and that always brings methionine, and that ensures that your first amino acid is going to be methionine. The confusing thing is, I think, is that your chart uses AUG. All right, so your chart, what codon stands for what amino acid, uses the mRNA. Why we use, say, AUG stands for methionine is because the only tRNA that can bring methionine, or that can bind with this, is the tRNA with UAC, and that only picks up methionine, so therefore AUG stands for methionine. Okay, so let's continue on here. As tRNA reads the mRNA transcript and brings an amino acid to the ribosome and adds it to the growing polypeptide. So that's what it says. So this tRNA will come over here. So the UAC will come over here, bring with it methionine, and we have our first amino acid there. So then let's look at this. Let's say the second codon, let's say this tRNA molecule has AAG, all right, as the anticodon. What I'd like you to do is to figure out what amino acid this tRNA would bind to and would bring to build a protein. So what amino acid would that tRNA molecule with the anticodon AAG, what amino acid would it bring? I'm asking for that tRNA molecule with the anticodon AAG. What amino acid would it bind to?
Or amino acid. So it's even a step back maybe primary. So the opposite of this would be so that's the opposite of that.
What do I use to figure this out? How do I figure this out? I use the chart. How do I use the chart? So my anticodon is AAG. Am I using AAG? No. All right. So the mRNA that this tRNA would bind to would have a codon of, let's pretend like this is the next one, UUC. All right, what binds with this? So therefore, we're going to use the codon of the mRNA that matches up with that tRNA in our chart. And so when you look at UUC, you get phenylalanine, PHE. Right? All right? So you have phe there. So that would be the second amino acid in our chain. And then we would look at the third, and so on and so forth, and we can build our polypeptide. So in this particular case, in the big picture here, the next codon would be UUC. We get the next tRNA molecule with the anticodon AAG, brings phenylalanine, and now we have our first and our second amino acid in our protein chain. All right, and so what we're going to do is continue on with that until eventually we reach what's called a stop codon. So tRNA is about 80 nucleotides long and full of complementary structures that hydrogen bond to one another, giving it that three-dimensional shape that I said looks like an L. And so we already talked about this here. From this, the anticodon loop comes out of one end. So this is the loop that they're talking about. So it's drawn like this on ours. And it has the 3' end that has the amino acid attachment site. All right, so let's look more specifically about what's happening here in this slide. Did I go too fast? Yes. So in this process that we go from picking up the amino acids to getting them in the right order to build your protein, there's two steps. There must be the correct match between the tRNA and the amino acid, so matching them up here, and the correct translation of the mRNA molecule, meaning that the amino acids are put in the right order to build the right polypeptide chain. So let's look at that correct match between the tRNA and your amino acid. So each amino acid gets joined to a tRNA by an enzyme. You do not need to memorize the name of this enzyme, amino acyl tRNA synthase. But synthase makes something. It's an enzyme. It's making it. What is it up to make? It's making a tRNA molecule bound to an amino acid. All right, so that's kind of what looking at it. There are 20 of these, one for each amino acid. Each enzyme catalyzes the attachment of the amino acid to tRNA. This was a typo, you have to fix this. The activated, instead of amino acid, it should be tRNA, is now ready to deliver the amino acid to the growing polypeptide chain. So we'll look at this picture. Here's this enzyme. Remember, enzymes have an active site. This enzyme has, part of its active site, a spot for an amino acid to bind to, And this part right here is a spot for tRNA to bind to. And so that's its active site. That's why there's 20 of them, 20 different enzymes. There's 20. Because why? Because there's 20 different types of amino acids. Each amino acid has a different shape, so each enzyme has to have a different shape to bind with that amino acid. So two things help to do this process. For the tRNA to pick up the appropriate amino acid. The enzyme helps, and so does ATP for energy. And so, ATP is used. How do we use ATP? We break off a phosphate. Oftentimes the molecules are phosphorylated. So here's your amino acid. It has become phosphorylated. And then your tRNA molecule, not bonded to anything, binds to the active site. And if you remember with enzymes, there's that induced fit hypothesis that once the substrates, which is the amino acid and the tRNA bind, they kind of I use the analogy of it hugging, all right, can orient the molecules so it's easier for them to bind. And so now we have a new bond here between the amino acid and the tRNA, and now we have this activated tRNA molecule right here. All right, notice that. So that's what happens in the process of it picking up the amino acid. So an enzyme is involved in that. Enzymes are not used up, so it's ready to go, and it can bind to another amino acid, and so on. So now the tRNA has the right amino acids. That's how they get bound to them. So then we have to bring them to the polypeptide chain. So the tRNA must correctly match up the tRNA anticodon with an mRNA codon. There is not a 1-to-1 ratio of tRNA molecules with mRNA codons. Some tRNAs can bind to more than one codon. This is called the Waddle effect. So I spoke briefly about that a little bit when I was in the notes in the video. So before I talk about this Waddle effect here, let's just look at this overview picture. So here we have our tRNAs that aren't bonded. Here, this is what I was trying to draw kind of over here. That's what this picture is showing you. Here are your amino acids from your diet, and your tRNA molecules pick up the appropriate amino acids. So every tRNA picks up a different type of amino acid depending upon what anticodon they are. And then at the ribosome, which is the brown thing here, this is your mRNA strand. Then your mRNA strand is read in groups of 3 of codons, and the tRNA molecules with the appropriate anticodon brings the appropriate amino acid, and you can build a polypeptide chain. So let's go back to this Waddle effect, that some tRNAs can bind to more than one codon. This Waddle effect enables tRNA to bind differently in one of its base pairs. It's usually the third base pair. This is why codons for some amino acids differ in their third base. So for instance, if we go back to that chart, like if you look at proline or pro, all right, so you have, so an example here would be proline, the codon could be CCC, could be CCG, could be CCA, or CCU. And all of these are codons that stand for pro or proline. This is the mRNA codon. I just copied it right from that chart. And that's what I mean by multiple codons can code for proline. And you can see that the first 2 bases are the same. What differs is that third base. So it doesn't seem to matter what that third base is for this. It could be a C, a G, an A, or a U. So that means that what happens is if you have a tRNA molecule, so if this is the mRNA, oops, throw that red marker away. You have a tRNA molecule that corresponds with CCC, so it's going to have GGG, goes with that, and that would bring, CCC stands for proline. But what they found is that these tRNA molecules, as we said, over here that some tRNAs combine to more than one codon. That this has a G here, but notice that CC, CCG, CCA, CCU. So they're found that if you have CCU, that this still brings proline, that it doesn't seem to matter what that third base is for that tRNA molecule, which is why we have multiple codons that code for the same proline and not just one, okay? Is that alternating of the third base there. That's why they say it's redundant, the genetic code, okay, because of that. It's the third base. All right, so our tRNA molecules pick up our amino acids, and then as I drew up here, they have to take them to the ribosome. So let's spend a minute and talk a little bit about the structure of the ribosome. So hopefully you remember from before that they're the sites of protein synthesis. But one thing about them is that they have a large and a small subunit. And that large and small subunit are comprised of RNA and protein. And the RNA is what's called ribosomal RNA. So I'm going to teach you a third type of RNA, adding on to this picture here. So here's still your DNA molecules. So some genes make mRNA to code for polypeptides. Some genes code for making tRNA. Are we okay? Oh, there's a lot of rumbling. All right, so we have the gene here. This gene doesn't code for making a protein either, but instead of coding to make tRNA, this gene codes for making rRNA, which is ribosomal RNA. And so, so again, not all genes code for making proteins. They make these other types of RNA. So if my board is the nucleus, in the nucleus, and when you look at the nucleus underneath the microscope, this is the nucleus here, you see a large dot in it. Do you guys remember what that is? There's there's a main structure inside the nucleus. It's called the nucleolus. And so, nucleolus. The nucleolus, when you memorize what it does, one of the things it does is make ribosomes. So what happens is our chromosomes and our DNA, all right, in our nucleus, so that's us in the nucleus, some of the genes make rRNA. The rRNA goes to the nucleolus, and we make the subunits. This is the large and the small subunit of the ribosome, and this subunit is made up of rRNA. So this little squiggly thing is rRNA. And this one is also made up of rRNA as well. And so that's what that means, that it's ribosomal RNA. We make ribosomes from rRNA. And so that's going on, big picture here. So that's going on right here. Here's the nucleolus in here. So we make the rRNA and make the ribosomes, and then it leaves the nucleus, and then our ribosomes these are ribosomes in the cytoplasm. So they leave the nucleolus and go out. That's how we make the ribosomes. So rRNA genes, so this is what I just drew here, rRNA genes are found on chromosomal DNA and are transcribed and processed in the nucleolus. They are assembled and transferred to the cytoplasm. The large and small subunits form one large subunit only when they're attached to mRNA. So normally, the ribosomes are detached. You have a small and large subunit, and they only come together when they're going to do protein synthesis. So let's look at the structure here. The structure of ribosomes fit their function. They have an mRNA binding site, a P site, an A site, and an E site. And I'll talk about what show you a picture in just a second to explain this. The P site holds the tRNA carrying the growing polypeptide chain. The A site holds the tRNA carrying the next amino acid to be added to the chain. And the E site is the exit site, before tRNAs leave the ribosome. Each of these are binding sites for the mRNA. So let's see. So make sure for just a second here. Oh, and on this, it's the blank is mRNA, but then you see how there's an rRNA down there? That rRNA was supposed to be the title of this next slide, like, which is the next. Did you see that? Like, all right, so you can cross off rRNA, and then you can either put the title above that bullet or leave it. So this rRNA making up ribosomes actually is the most abundant RNA in the cell because we have so many ribosomes, and every ribosome is made up of rRNA. So it's way more abundant than mRNA or anything like that, although most people have only heard of mRNA. Okay, so we're going to look at specifically now at the ribosome. How do these tRNAs bring the amino acids to hook together? And there's three stages. Initiation. So when the mRNA comes to the ribosome and you have a start codon, that's the initiation process. And so the first tRNA brings the first amino acid, and that's initiation. Elongation is then the reading each codon in different tRNA molecules bring the next amino acid, so you're elongating your polypeptide chain. And eventually termination, you terminate it, so instead of a start codon, eventually on the mRNA, we reach a stop codon. All right, and that's how we know when to stop adding amino acids and when your polypeptide chain is done. And so this GTP energy source powers that. So let's look at that first step, initiation. Brings together the mRNA, the tRNA, and the two ribosomal subunits. And GTP is that energy source that brings the initiation complex together. So all these things together. GTP is like ATP, except it's guanine instead of adenine. But it's used the same way in that breaking off a phosphate and so on provides energy. So initiation brings together RNA, the two types of RNA, mRNA and tRNA, and the two ribosomal subunits. Look at here at the beginning. This is part of the ribosome. This is the small subunit. So in these cytoplasm floating around, you usually don't have a completed ribosome. There's the small and the large subunit floating around by themselves. They're not attached. So what happens is that when there's an mRNA molecule, the mRNA molecule binds to the small subunit. So here's your small subunit, mRNA binds to it. The subunit has a binding site for that to happen. And only when that happens does the large subunit come in. So therefore, we have a fully built ribosome, but that only happens when mRNA is attached to it. So then notice here, it says start codon. So here's your start codon, bringing with it the tRNA with the matching bases, the anticodon that binds to that with the appropriate amino acid. So your start codon, remember always AUG. So the anticodon is UAC, and that tRNA always brings that amino acid methionine. So therefore, notice here that we have with the large subunit that start codon is in the P site, it's the EPA. That's the order of these sites. Each site matches up with a codon of the mRNA. So notice that there are 3 bases in each site, EPA. So your first start is in that P site, and that's your first amino acid. So now we want to bring in our second amino acid. And so let's say that the next codon is, let's say it's ACG. So if the next codon is ACG in this picture here, what I want you to do is on this picture actually draw in a tRNA molecule, write in what anticodon it would have, and attach what amino acid it would have attached to it. So what would be that second amino acid? So the anticodon's gonna have the appropriate complementary cases. So what goes with an A would be a U, C would be a G, and G would be a C. So there's your tRNA. And then it's gonna bring with it the second amino acid that's gonna be in this particular chain. So what are we gonna use? Remember, we always use the mRNA in the chart. So we're not going to use UGC, we are using ACG. So then if you look at ACG, that's THR. All right, so THR is threonine. That's THR is that second amino acid. You guys able to do that? Yes, you have that second amino acid, right? Okay. So that's amino acid 1, amino acid 2, and so then how are we going to get to amino acid 3? We need to look at the next codon, and the appropriate tRNA will bring the next amino acid, and so on. And that's going to start being the elongation part. So we've started this elongation part. So let's look at this elongation, and then I'm going to come back to that initiation part here. So let's look at what happens. Let me erase this here. So notice here, we're going to start here and we're going to go around here. So here's your EPA site. Here's your tRNA. Notice here it has multiple amino acids attached to it as a growing amino acid chain. So this codon is right here. This is the next codon. So then in the A site, we have a codon here that has the tRNA with the appropriate anticodon coming in and hydrogen bonding with it, bringing in the next amino acid. Now, if you look from this picture to this picture, notice this tRNA molecule in the P site no longer has any amino acids attached to it. Where did those amino acids go? They went, and this bond was broken, and they went and bonded to this amino acid here. So this is the new bond right here that was made. So all the amino acids that are in this site get transferred to the amino acid on the next tRNA molecule. Now what happens is, if we look from here to here, notice this arrow. What does this arrow mean? It means that the mRNA shifts to the left by one codon. So what do I mean by that? So here's the codon here that matched up with this tRNA. This is the codon here, and this is the next codon. So what happens is, it would be like if I were to actually take this mRNA and pull it and just shift everything to the left. So this tRNA now is no longer, its codon, and this tRNA is no longer in the P site, it's now in the E site. And this tRNA molecule has done its job, it's brought its amino acid, and so in the exit site, it exits. It's done its job, and this amino acid can go bind to another amino acid. So this can be reused, so that means it's being reused, that tRNA molecule. So it's done its job. So now the codon that was in the A site with this tRNA molecule is now in the P site. So this is why the P site we wrote has the tRNA with the growing amino acid chain. And now the next codon is in the A site. What's going to happen now to bring the next amino acid? The tRNA molecule with the appropriate anticodon that will bind to that brings the next amino acid to the chain. So it comes over here and binds. What's going to happen here? This bond is going to break, and it's going to attach to this one here, and this cycle continues, and it keeps on shifting to the left and shifting to the left. And so let's go back to this picture then. So this, let me ask you this. So here we had, what did I say, ACG I think it was, and so we had UGC, and we had threonine. All right, so that's that. So according to what we just looked at with elongation, what's going to happen next after this? So I had you draw in the tRNA with the appropriate anticodon, the appropriate amino acid. What's going to happen next in this process? There's the first amino acid, second amino acid. What's going to happen to this bond? Emily?
It's going to break and attach to the other one.
That's right. This bond breaks. Methionine is going to attach to threonine. And now we have a dipeptide. First amino acid, second amino acid. What's going to happen next? What happens to the mRNA? Shifts. So now look at this tRNA will be in the E site. It's done its job. It brought the first amino acid. It's going to exit and leave. This tRNA is going to be in the P site. It has the growing polypeptide chain, the first and second amino acid. And then this next codon that codes for the next amino acid will be in the A site. What's going to happen here? Another tRNA molecule will bring the next amino acid, and then this bond will break and attach to that, and it continues on and on and on. Until we reach a what? Until you reach a stop codon. So that's the termination part. So termination occurs when a stop codon on the mRNA reaches the A site within the ribosome. Release factors then bind to the stop codon in the A site, causing the addition of water to the peptide instead of an amino acid. And this signals the end of translation. So we have the end of the protein. So if we look here, so we have our growing polypeptide chain. When the next codon here is a stop codon, and there are 3 stop codons. When you look at your chart, stop codons, they'll just say stop. Unlike start, it says start or methionine, because start actually codes for an amino acid. Stop does not. So therefore, stop literally means stop. We're not going to add any more amino acids. And so what does bind there is this release factor. The release factor, says here, causes the addition of water. When you add water, why do we add water? What happens? Hydrolysis. We break the bonds apart. So we added OH to one molecule and H to another. So what's happening is that instead of adding another amino acid to the chain, it's adding water, which is going to break this bond here. And so now if you look at here, the tRNA no longer is bound to the polypeptide. The polypeptide is finished. And there's your free polypeptide, and everything dismantles, and the ribosome goes back into the large and small subunit, and will stay that way until another mRNA molecule binds. All right, and so that is your end of protein synthesis. All right, and your polypeptide. Okay. I guess I'm gonna do this one last slide. Hey, I let you out early, so. As the polypeptide is being synthesized, it usually folds then and takes on a 3-dimensional structure. Remember that proteins aren't just chains of amino acids, they have a 3D structure. So we have also post-translational modifications after translation that are required to make the protein function. We can add certain chemical groups, fats, sugars, phosphates, remove certain chemical groups, and sometimes proteins have to be made by, or have multiple polypeptides to make the protein come together. All right, and so you guys are. I'm finishing the notes on Monday, and our test is still on Tuesday. That's just how it has to be. Okay. All right. But what I'm going to do is this weekend, because we have to finish the notes, is I'm going to do an overview review video of everything so that you can use that as a review. All right. We won't have a whole class period to do that. Okay. So,
Like your socks and shoes, Kate.
it'll probably be uploaded tomorrow.
Bye.
Steady that pencil and move your watch.
Hey, did you ever find the answer to that question about what to do? Like,
Yeah, Allison asked for counselor and she said that she would write AP in there and then draw a row to AP environmental science and then they found that it's only if that.
Gotcha. Yeah, that's good to know for you.
See you, Miss Walker.
Have a good weekend. Oh, I forgot about my video.
You got a video, Sydney? Good.